we're gonna start from the preclinical side and thinking about developing therapeutics for Alzheimer's disease. Why isn't this? There we go. So the first lesson is that drug discovery is not easy. I mean, it takes a long time. You start with hundreds, tens of thousands to millions of compounds that you might screen, you know, developing a lot of lead candidates that go through preclinical evaluation to get to some clinical candidates that you might conduct a trial with. And at the end of the day, maybe you get a drug or not. You know, and for Alzheimer's disease, it's mostly been not getting drugs. And this process, in addition to taking a decade, might cost $1 billion or more. You know, outlined here are kind of 3 different strategies for drug development. The only one that's really very cost-effective or fast is drug repurposing, where you take a drug for one condition and try to repurpose it for another. But that's also got its challenges. But you know, we're really going to just focus on mouse models and barriers, and you know, some of the reasons why the field has really struggled, as opposed to, you know, a comprehensive discussion. This would of course take hours to days to, you know, cover everything. But you know, I think that, you know, the main barriers to developing therapeutics preclinically are that our animal models don't really reflect amyloid-dependent tangle formation driving neuron loss. We don't have good cellular models for the pathology of Alzheimer's disease forming in a dish. We still have a pretty incomplete understanding of all the risk factors. There's you know, kind of confusing interplay between the various AD-related dementias and Alzheimer's disease that includes, you know, not understanding what exactly the toxic species might be. You know, clearly drug development to date for Alzheimer's disease has really had at least for the past 20 years, has been almost solely focused on the amyloid cascade hypothesis, which we'll talk more about. And the genetics have clearly highlighted, you know, the importance of glia and neuroinflammatory contributions, but there really hasn't been a lot of effort so far in the neurotherapeutic space on that. And generally, we tend to disregard the biology of aging as the main driver of Alzheimer's disease, but it is certainly the, you know, the biggest risk factor. And, you know, probably most importantly, we seem to be repeating our failures over and over again. So we're not learning from our past mistakes. Before we get into the, you know, one vignette on Alzheimer's disease drug discovery that I think is illuminating, I'd just like to talk about a different neurodegenerative disease and how some things there went right. And that disease is spinal muscular atrophy. And the thing you need to know about spinal muscular atrophy is it's an incredibly severe neurodegenerative disease that's caused by recessive mutations in the SMN gene. Depending on the expression levels of related SMN2 protein in cases, you can have neurodegenerative disease that has onset from, you know, infants to adults, but the infantile form is extremely severe, typically killing kids, you know, at the age of 2 or 3, sometimes younger. And the thing that really made a big difference for SMN in the development of therapeutics is that relatively, you know, recently, about a decade ago, it was shown that overexpression of SMN2 could rescue loss of function of SMN1. And so there was a therapeutic idea and a target in mice that seemed to be pretty applicable to humans. And, you know, if you want to read about this, there's a very nice Nature paper from Adrian Kraemer's group where they show that by manipulating SMN2 splicing, they can completely rescue the neurodegenerative phenotype in this mouse model. And this mouse model really is a very good replication of the human disease. And so, you know, in the course of, you know, 5 years, we went from therapeutic idea to ASO that rescued SMN2 splicing and humans. So, and in fact, we don't have one drug, but we have 3 drugs, and they're all essentially functional cures. I mean, if you go look at the videos online of, you know, little kids before and after the start of therapy, they're learning how to walk, they're doing all the things that, you know, you might expect a normal kid to do. They have, you know, great motor function. So this has really been a huge win for the field. And, you know, 20 years ago, you'd have never predicted this is the first disease that we're going to cure of a neurodegenerative type. And, you know, so I think the big difference between this and a lot of other neurodegenerative diseases is they had a mouse model that really reflected the human disease, and they were able to cure that model and then, develop therapies based on that model that worked in humans. And in, 5 years, we went from no approved drugs to 3 drugs that are all, apparent cures or near cures, very, very potent therapeutics. So for Alzheimer's disease, the target of choice in the field, more than 20 years ago now, was amyloid. It was found that a portion of the APP protein was processed to form the main component of plaques. This is biochemically defined, and then subsequently genetic mutations that influence APP processing in the form of mutations in the APP gene or the processing proteins presenilins 1 and 2, or all cause early onset Alzheimer's disease. And so the field was, very enthused about this and came up with this amyloid cascade hypothesis where misprocessing of amyloid was the driver of Alzheimer's disease. And then the race was on to build mouse models, and there's a whole bunch of mouse models where you can overexpress human APP with these different mutations and get a whole bunch of plaque deposition. And so, you have one of the 2 hallmarks of Alzheimer's disease, amyloid plaque, all over the brains of these mice that causes some cognitive problems for the mice that are easily detectable and some hyperexcitability of neurons. And so this was viewed as a good mouse model of Alzheimer's disease, and drug discovery began. What these mouse models all really lacked was loss of neurons, tau pathology, brain wasting, premature death, the sort of things that also go with Alzheimer's disease. So it was obviously an incomplete model of the disease, but the plaque pathology is very, very convincing looking plaques, at least in some of the mouse models. And so drug discovery ensued, and it wasn't very long before people started to find approaches that could cure these mice. And I'm only going to talk about one approach because I think it really did inform us very early on about the amyloid cascade hypothesis and what we should have been thinking about, sooner than really the field is just now starting to raise these sorts of questions. But I think that the data addressing all this has been out for more than a decade. So you take this mouse model with all these plaques. If you immunize them against the A-beta, the human A-beta peptide, you can clear the plaques, and the mice are cured of their, their plaque disease that was viewed as a good model of Alzheimer's disease. And that rapidly led to a clinical trial run by a company called Elon, and the therapeutic agent was AN1792, which was an active immunization against A-beta peptide. And the phase 1 trial went very smoothly. It seemed to be safe, went well. They started to immunize Alzheimer's patients in their phase 2 trial, and it was stopped, midway through the second round of immunizations because of adverse events. And these are not like mild adverse events. We're talking about encephalopathy and premature and death. So, 6% lethality in that trial is really, kind of the nightmare scenario when you run a clinical trial. Nonetheless, I think the trial could be very informative for the field. And here's just a little bit of the design. And I'm not trying to steal Elaine's thunder. She's got all sorts of trials to talk about. But I just want to talk about this one because it's all predicated on this, amyloid mouse model. And so, the trial was stopped. Obviously, this was not going to help patients in any obvious way, and it was, putting a bunch of them at risk. So it was obviously the right thing to do. But a bunch of these people were followed for quite some time, through the course of their disease. And I think there's a lot of information that can be gleaned from that. In fact, a couple papers have been published. But one of the things that was observed was that, they studied the patients that had a, measurable antibody response against A-beta, and the intervention did not seem to change the course of their cognitive decline. And obviously that's the thing you want to see first when you're looking at something that's going to intervene in Alzheimer's disease, is you want to preserve some cognition, and that didn't seem to happen. Didn't seem to change their survival. And, most importantly, it worked molecularly. It cleared the plaques. That was what it was supposed to do, and it did. There are no plaques in these high titer responders. Tau is still there. The neurodegeneration went on, but and obviously this is 10 years after the trial was done to follow up to get this data. But, you know, and it's not a huge number. I think it's like 30 cases or something, but it's pretty clear the intervention did what it was supposed to do in the sense that it cleared plaques, but it didn't seem to intervene in the dementia. And, you know, this should have told the field, we need to think about this hypothesis more. But instead, the lessons that were really learned by the field was we need to figure out a way to clear plaques without, you know, causing this neuroinflammation, and that's going to solve the problem. And, you know, in the patients that didn't have an encephalopathy, you know, some of these high titer responders that they did autopsies on, there was no increased neuroinflammation. It wasn't that. It was, you know, just that clearing plaques is not enough. So, you know, I think the field has been over-focused on this idea that clearing plaques is going to cure AD. And, you know, the idea now is, oh, we just need to go earlier. And maybe that's true, but I think that experiment has already been done in the phase 1 trial that Elon did, because they immunized a whole bunch of people against A beta before they had any symptoms. They were young, healthy folks. And the question is, are any of them going to get Alzheimer's disease? It's a very long-term study, obviously, but you know, I think that might ultimately answer the question, you know, for once and all. But I think, you know, those people were in their late 20s on average 20 years ago, so they're in their late 40s now. We got another 2 decades to wait probably, and then we should find out more about that if anybody's following them. I don't know if that's true. Anyhow, I digress. The clear thing that these studies showed is that tau pathology is important for dementia. Removing plaques alone is probably not going to be enough, and that, you know, there seems to be some sort of synergism between tau and amyloid driving neurodegeneration and Alzheimer's disease. That may be that, you know, amyloid is first, triggers the tau pathology, and then once tau pathology has been triggered, there's amyloid is dispensable for the neurodegenerative cascade. If that's true, then going super early might help. But, you know, I think that this is one problem in the field, is this focus on amyloid. Another is there's not a lot of diversity in the ideas that are going forward. You know, this is the most recent classification I could find about, you know, what trials look like. And, you know, a striking number of the planned trials are still focused on the amyloid cascade, despite, you know, more than 100 failures. Just, you know, over and over, the field has failed targeting amyloid, even though they've had molecular success in many instances where they've actually intervened on amyloid. They haven't done a lot for cognitive improvement, which is concerning. So last slide is just the take home. I think the field needs better preclinical models of AD, where you capture not only amyloid plaques, but neurofibrillary tangles, and most importantly, I think neurodegeneration. If your model doesn't have good neurodegeneration, it's probably not a great model of Alzheimer's disease. I think also overinterpretation of animal studies can misguide trials that are being done, and that, you know, we probably need to diversify our therapeutic targets for AD. You know, it's not as obvious for Alzheimer's disease as it was for SMA what the target should be, and I don't think the field has really arrived at those neuroprotective targets yet. But, you know, hopefully that can come in the near future.